Welcome to the most exciting session of the symposium, where we highlight our early career investigators. There is an order to this session. I think we start with the basic science talks, and then we move to the clinical talks. You'll be hearing from postdoctoral research fellows, clinical fellows, and even graduate students. Our first speaker is Collier. Right? She's a postdoctoral fellow in the Department of Dermatology. She is the recipient of the MCHRI Postdoctoral Support Award, which provides 2-year salary support. Application deadlines are around the fall of every year, so think about it for next year. And she will talk about genetic mechanisms and ectodermal dysplasias.
Hi, everyone. Thanks for coming back after lunch. And thank you so much to the organizers for giving me this opportunity to share some of our work with you today. So I'm Annie. I'm a postdoc in the oral lab in dermatology, and today I'll talk to you about how we've used a new proximal proteomics technique to reveal what we think is a new regulator of embryonic development. So as you can see from this stock photo, during normal development, there are obvious differences that arise in terms of the appearance of skin, hair, facial features, teeth, and so it's no surprise that when you have a disease that affects these tissues, such as ectodermal dysplasia, there's a baseline heterogeneity that exists there as well, and that makes these diseases quite difficult to classify and prevent and treat. And that heterogeneity is evident in the fact that there are over 180 types of ED that have been identified in the literature, and these range from something mild like a hair and skin defect to something more severe like a cleft palate that requires surgical intervention. And so our lab proposes that if we can study the molecular and cellular mechanisms behind the development of these tissues, we can better understand how to prevent and classify the disease. And so obviously we're from the Department of Dermatology, so our primary interest is skin, which comes from the surface ectoderm subset of the ectoderm germline that arises from early stem cell differentiation. And in the oral lab, we've developed a in vitro protocol where we can actually make surface ectoderm in a dish by differentiating human embryonic stem cells with retinoic acid and BMP4. And we can take these cells all the way to keratinocytes by adding keratinocyte media. And previous work in our lab has done a full transcriptomic and epigenomic characterization of this early differentiation process. And what we found is there's a subset of transcription factors that we think are really important to drive not only gene expression changes during this process, but opening and closing of chromatin or chromatin accessibility. And so what we wanted to figure out next was what is the exact mechanism of these factors in opening and closing chromatin. And particularly, I'll focus on the AP2, GATA, and P63 transcription factors. So the way that we're studying this is through a really great technique that was developed recently by the Collier lab, also in dermatology. And if you're familiar with the bioID proteomics technique, this vastly improves the specificity and the speed of that technique. So what you do is you take a BASU, which is a promiscuous biotin ligase, and you fuse that to your protein of interest, in this case, one of the 3 transcription factor families. And when inserted into your cell of interest and you add biotin to the cell culture media, this ligase will biotinylate anything bound to your bait protein or anything in the small radius nearby. So you're capturing a really specific interactome of whatever protein you're interested in. And then you can simply purify these proteins with streptavidin and perform mass spectrometry. And so what we hypothesize is that by using this technique, we can identify the interactomes of our factors of interest and possibly identify the chromatin remodeling complexes that they're working with. And we were able to identify these interactomes for these 3 different families. And what you can see is that each member has its own subset of interacting proteins, which makes sense because these are very different proteins at the structural level. They have very different domain organization as well. But there is quite a bit of overlap between the interacting proteins of all 3 families, and those proteins are enriched for transcription factors and chromatin remodeling complexes as we expected. And we're actually able to fully resolve entire known chromatin complexes, such as in this case the NERD complex and the BAF complex. And the mechanisms of those interactions are currently under investigation in our lab. But kind of a side observation we made when analyzing this data was that there's a small subgroup of DNA binding proteins that were highly enriched in all of our interactome data sets. One such protein is called AT-hook DNA Binding Motif Containing 1, or AHDC1. And like I said, these proteins are really largely uncharacterized. There's no mechanism. We don't really know what they do. In the case of AHDC1, it's been recently associated with a disease called Shaw-Gibbs syndrome. And this manifests as dysmorphic facial features, intellectual disability, and a broad spectrum of global developmental delay. So it's affecting a lot of tissues that arise from the surface ectoderm or the ectoderm. So all of these clues together pointed us in the direction that it's probably important in the early specification of the surface ectoderm. So to figure out what this protein does in our system, we used CRISPR-Cas9 to delete the gene in human embryonic stem cells and then performed RNA-seq. And what you can see is there are massive changes in gene expression, both at the positive and negative level. And a lot of the genes that are changing when you delete AHDC1 are genes that are associated with nervous system development as well as epidermal development, which are two tissues derived from the ectoderm. And since we found this protein to be associated with our transcription factors of interest, we wanted to know, are they co-regulating genes with our factors? Is that why they're binding to each other? And if you look at just a principal component analysis of knockout cells that we generated for each of the three factors, as well as AHDC1, you can see that there's the strongest correlation between AHDC1 and the GATA family. And there's quite a striking overlap between the actual genes that they're each regulating. And further confirming this correlation is the fact that the majority of genes regulated by AHDC1 actually have GATA physically bound to the promoter of that gene or bound somewhere far away and physically looped in by long-range chromatin contacts. And what makes GATA different from the other two families of transcription factors is that it contains a zinc finger. This is a very abundant protein domain in the genome, so we thought maybe this is how the interaction is mediated, and maybe GATA or AHDC1 is a general interactor of other zinc fingers. So we went back and also performed proximal proteomics on AHDC1, and we found that it does, in fact, predominantly interact with zinc finger-containing proteins. In fact, over 50% of its interacting proteins contained a zinc finger. These are primarily C2H2 type zinc fingers or Crab domain-containing proteins, which tend to be repressive in nature. So this, along with other data I won't have time to show you today, led us to this model that AHDC1 might be acting as an adapter protein between a site-specific zinc finger-containing transcription factor and a chromatin remodeling complex like the NERD complex. And all of this is why we think AHDC1 is important in the ectoderm. But what we didn't know yet is we didn't have a functional link between the gene and the phenotype that we think might be happening in patients. And there's currently no mouse model for AHDC1. So we first took our human embryonic stem cells and differentiated them all the way to the keratinocyte lineage. And what we saw, just by morphology, is that compared to the wild type, which we usually get these nice little clusters of functional keratinocytes surrounded by contaminating fibroblasts, and the AHDC1 knockout cells kind of stalled at this early surface ectoderm morphology and didn't really go any further, and they just started to die around the edges of the colonies. So it seems like they can't make it all the way to the lineage that we're trying to get them to go towards. And then finally, to really figure out what this gene is doing in vivo, we took advantage of a CRISPR pronuclear injection, which gives us a spectrum of mosaic mouse embryos that we can characterize by the expression of the AHDC1 protein. And these mice are E18, so we extracted them the day before they would be born, essentially. And you can see as you lose AHDC1, you're getting global developmental defects, and that includes skin defects, there's hypovascularization, the eyes are open at birth, there's omphalocele, and they're obviously not viable. And if you look at the skin specifically, there are a lot of differentiated layers that are missing. There seems to be a basement membrane defect, but possibly some sort of blistering phenotype. And we can actually take the skin from that E18 mouse and graft it onto an immunocompromised adult. And what you see in the mouse that's deficient for AHDC1 is that the hair fails to cycle properly and it doesn't form a full coat like what you see in the wild type. And this is actually really similar to what you see in the GATA knockout mouse as well. So taken together, we've used a multi-dimensional cellular biology approach combining proteomics, genomics, and epigenetics to detail a mechanism of a new protein, which we think could potentially be a new link to ectodermal dysplasias, as well as possibly some undiagnosed intellectual disability. So we think that this could be potentially a new marker for this disease and something that could be used, like we talked about a lot today, in like pre-screening of children and help classify these really heterogeneous diseases. So just quickly like to thank everyone in the Oral Lab, especially the people I bolded here that helped directly with a lot of these experiments, and also the MCHRI for providing me with funding for my postdoc. Thank you.
So the phenotype of the mice is more severe than I would say, the surviving patients with Collier syndrome.
Yeah.
And I imagine it's because the surviving patients have mild mutations, hypomorphic, you're not really seeing real knockouts.
Right, so all of the patients that have been identified with the mutation are heterozygous, and they're all de novo mutations, so they probably have some degree of mosaicism.
So it's a haploinsufficient phenotype.
Exactly.
So even at that, you can say
Yeah, so yeah, we don't really know what would happen if both copies were deleted in an actual human.
probably would make it.
Yeah, probably not.